Let's now take inventory of our Wikipedia signaling page. So now we've added the Wnt-beta-catenin story to our regulation. We've also added integrins activating focal adhesion kinase and therefore the MAP kinase pathway, also through AKT, promoting survival as they do. So now we're ready to get to this business here, which is the arrows that lead to something called cell proliferation, which is just another way of saying the cell cycle. So we'll talk about the regulation of the cell cycle as a decision, a regulatory phenomenon, and somewhat as a physiological event. But mostly we're going to be concerned with the control of the cell cycle, and that's where these cyclins and other components are going to come in. And that we'll talk about in the context of multicellularity, the need to balance the drive to proliferate with two other things: the inhibitory processes that keep that from happening when it's not supposed to, and also apoptosis, which is a way of ending the existence of the cell in such a way that it has the least negative effect on neighboring cells. So in order to make sense of this term cell proliferation and talk about it in some sort of meaningful way, it's time to get down to the business of the cell cycle itself. And these diagrams, of course, have been around forever, probably since kindergarten. You've seen them. And they're good for what they do. What they don't do particularly well is indicate that what's going on is one cell becoming two. So you have to just know how to translate this circular diagram into a binary fission, a division event. So that, for example, cytokinesis is a separation of the cytoplasm, so one cell is becoming two. In the S phase, the S is the synthesis of DNA, so the chromosomes are being replicated, the genome is being duplicated in that process. So the stage for that cell division, of course, is set during mitosis. In the case of cells that are undergoing asexual reproduction, so the chromosomes in this stage condense and obviously separate. So for many years, there was the M phase, which was visible, and then there was everything else, which was called interphase, during which nothing obvious was going on whatsoever. You needed biochemistry to prove that the DNA content was doubling in the S phase, so that came much later. The phases in between were referred to as gap phases, and they were sort of question marks in terms of what happens. There is, however, a very, very important invisible phase that occurs somewhere right around in here for us, because we're concerned about the control of the cell cycle and whether to undergo it in the first place. If you took a census of all of the cells in your body right now, the huge majority of them will say, I'm not currently undergoing cell cycle at the moment. And that's a reflection of the fact that most of the cells in a multicellular organism divide to a point and then stop, and then they are either officially more or less permanently paused in G1, or in some cases we indicate that they are in a sort of a distinct off-ramp from G1 called G0, or in a minority of cases they might be paused more or less permanently in G2. So we need a regulatory mechanism that takes all of those things into account. We've got to establish for the orderly, sequential activation of events that lead always from M to G1 and never from M to G2 or from M to S. So we need a unidirectional process in which no states are skipped. And we also have to provide a means of pausing and then of proceeding through the mechanism if the situation requires it. So over here is just a slight elaboration on this diagram with a nice little switch mechanism sort of built into it. So this provides, I think, a very nice visual depiction of the unidirectionality of the process. If you just in your head imagine a little ridge right here, a little berm, a little something that switch has to go over, then everything I think would be pretty much perfect. So in that case, what happens is the cycle continues until you get to that bump, and then it stops. And it requires some additional input in order to get the switch over that bump. Once it's over that bump, it's going to go all the way around until it gets to that bump again. That's the way to think about the cell cycle and the contribution of growth factors, because they are the things that provide the push to get that switch over that bump. So if we think of terms if we think of the cell cycle in those terms, then we can identify that particular location where this hypothetical bump exists. And depending on what type of cell we study, we would call that start. That's what it's called in yeast. And restriction point in the case of us, mammals, mice, etc. So to elaborate, we have to have a regulatory mechanism that accounts for the behavior of cells with respect to start or the restriction point. And it would be nice if that mechanism also includes a description of how you can use it to have replication of the chromosome go on here, condensation of the chromosomes, vesicularization of the nucleus, separation of cytoplasms go on, the actual physical events associated with the cell cycle. So if that mechanism describes the regulation and also the physiology, then that would be great too. So I'm pretty sure if you didn't know already how the cell cycle is controlled, at this point in the semester you would just automatically guess it's going to be protein kinase controlled. And of course you'd be right. The protein kinase in this case are new ones for us. We haven't talked about them, but it's the cyclins CDK, cyclin-dependent kinase dimer, that does it. And maybe if I made you say, well, what else would be involved, and you start thinking in terms about the unidirectionality, irreversibility of the cell cycle, you would say, sounds like the proteasome and ubiquitination. And if you said that, you would be absolutely right. And you would have 90% of it down. So the rest of it would just be details. And much of the tale would be the nature of the kinase itself, or kinases themselves. And I'm sure that this is a review, but the protein kinases in question are these. So it's a characteristic two-subunit arrangement in which the catalytic activity that transfers the gamma phosphate from ATP onto other proteins is something that's called a cyclin-dependent kinase, because it's a protein kinase and it requires an additional regulatory subunit called cyclin in order to be active. So the minimal requirement for this thing transferring a phosphate group is the presence of its cognate cyclin. So just this situation by itself provides obvious means of regulation, because this subunit can be made to cycle through the standard ways by which proteins come and go inside the cell. So that, for example, the various phases of the cell cycle could be regulated by transcription and translation, such that gene regulation could lead to the the cyclic appearance of cyclins, and therefore the cyclic activation of cyclin-dependent kinase activity. So we could call that gene expression. For example. And then there are other ways of making proteins come and go, of course. So gene expression, protein synthesis would be the way that they come. The way they go would be proteolysis. So the flip side of that coin would be, the UPS or UPP. The ubiquitin proteasome system of, exam 1. So by taking advantage of normally occurring physiological processes and the central dogma, it's possible to create a system of periodic protein kinase activity that, works with the tides, basically, that works on a cyclic basis simply by having alterations in gene expression and proteolysis control it. So there is a cell cycle dependent component to that we have to make a little bit more specific accounting of, and that's shown in these diagrams from your text. First of all, here is the standard circular diagram from earlier in this video and a million other discussions of the cell cycle that I'm sure you've had. If we take this and basically linearize it, do a linear permutation of the cell cycle, that's this diagram. And I just took two of those and stuck them back head to tail so that we could talk about the transitions a little bit better. But in any case, you have to begin somewhere, so we could begin with that going over the hump part that I talked about at start. That would be right here in this diagram. And what's shown is a number of things. One is the appearance of the active form of the cyclin-dependent CDK. Down at the bottom, the position in the cell cycle where we're talking, and then a measurement of either abundance or activity. So the color scheme indicates the cell cycle, and the size of this peak indicates either, you can look at it two ways, either the amount of the appropriate cyclin or the amount of kinase activity that cyclin stimulates. So if we begin here prior to the start or restriction point, then both of those things are low. And what happens first is that a G1 phase cyclin builds up, and that takes us through the start point, at which time an S cyclin goes from essentially 0 to full strength early in the S phase, and that controls the kinase activity of the S phase cyclin CDK. So whatever it is that turns on DNA synthesis, that ultimately is turned on by phosphorylations catalyzed by this complex. So once we get to that point, everything is going to be fixed all the way through mitosis, unless there is something that goes badly wrong in the plan, like DNA damage or something. But let's say that does not happen, then the S-phase cyclin CDK continues to phosphorylate things throughout S and into the second gap. Sometime during that phase, which tends to be shorter than G1 in a sort of stereotypical cell cycle, then the stage is set for mitosis ultimately. That's what we think of G2 as being, proofreading and getting everything ready for cell division. So that's catalyzed by the M-phase cyclins. So by the time we've activated them, then one cell becomes two, and that activity plummets. So I have two things to say about that before I'm done. The first is that the cyclin-dependent kinase doesn't have to come in multiple flavors. So that, for example, yeast cells get by with a single CDK, but they have specific cyclins that correspond to each of the phases. So it's a little bit more complicated in humans, but even in humans, we don't have CDK for each of the phases. That's not necessary. What is necessary is that there are cyclins for the specific phases, and they go by different names. And in the case of the diagrams that we'll see in the next video, it's going to be important for you to distinguish some of those. Cyclin D in particular, that is the cyclin, the gene for which gets us through start. So the amount of cyclin D is of huge importance in terms of the physiology of the cell. What it does ultimately is just one thing, is provide for the expression of cyclin E, and that leads to the activation of the cyclin CDK activity that takes us into the S phase, and therefore we're going to go all the way through the cycles after that. Okay, so the periodic expression of cyclins goes cyclin D, cyclin E, and then once we get into the late G2, cyclin A and cyclin B take over and phosphorylate different targets as a result of their association with either the same or different CDKs, and then we have the cell cycle completed. So if you look at the appearance of these peaks, it's very often the case that you have a gradual increase and then a very sudden decrease in either the activity or the concentration of cyclins, depending on how that is being measured. So that gradual increase probably reflects a number of activating mechanisms, including gene expression. A very sharp decrease, a plummeting of activity that you see here and here, for example, that's a very good indication of proteolysis, that the cyclins are being allowed to exist in work, and then at some certain point, there's a specific signal that says, get rid of all of them now. So that's an indication of proteolysis. And then finally, there's this part right here. So if you think in terms of kinase activity, then you just plot kinase activity period, regardless of which cyclin CDK is doing it. Then you get a straight line going through the cell cycle, going through into M, and then dropping to 0 basically, and staying there until it goes up in, let's say, late G1, and then it stays high all the way through the next M phase. Drops right at the interface between M and G1 and stays down again. So what's going on here is basically the licensing of the cell cycle. It's a cock and fire mechanism when it comes right down to it. And what you need to think of in terms of making sense of that pattern is that, yeah, there's specific kinase activities and proteins are being phosphorylated. But while that's happening, there is a background activity of phosphatase that is continuously removing the phosphates from those targets. So as long as there's kinase activity, then you get some steady state level of protein phosphorylation. But after you have undergone that cell division that would be described here or here, then you don't have any more kinase activity. So all those phosphates are going to be removed because phosphatase activity continues across this gap. So what is happening is essentially a resetting of the cell cycle as a result of the absence of kinase activity, but the continued presence of phosphatase activity. So that's not going to make a whole lot of sense at the moment, but just sort of keep that concept in mind, that for all these kinase activities, there's a corresponding phosphatase, and there is one point, and only one point, in the cell cycle where phosphatase absolutely wins, and that's right here. That's that late M phase, early G1 phase.